Good afternoon, everybody. I am delighted to be here. This session, when the organizers invited me, was Smart Lab, the sort of lab of the future, and I'm going to tell you the story of my own particular lab, and mostly as an example of what it takes to actually keep a lab as a smart lab of the future, and really a journey of, let's see if this is working. There we go. Of what I've done for 15 years. And this is basically a very simple idea, but a very powerful one. And it's the idea that every living thing that we encounter, ourselves, plants, animals, have within them this code, this genetic code, like software, that actually encodes all of their properties, all of the cells that we have, the structure. And if we could only read that code, we could access recipes, nature, we could access all of this biology. And then as we got better at reading this code, we could actually start to engineer it. We could actually start to modify things, actually start to use biology to do interesting things. And this is what my lab has done for the last 15 years. And it's an interesting story that I'm going to tell you about. So I started out as a petrochemical engineer. I didn't really love it, and I was lucky enough to, so I quit, and I was lucky enough to go to MIT. I did a PhD and MBA there, and while I was doing my PhD, I was even luckier to be part of the group that was doing the Human Genome Project. So this was kind of, at least in large-scale biology, one of the kind of the first labs of the future. So everything you see there, we had to invent, we had to create. Every machine, every robot that's in that picture around 2000, none of them existed. You couldn't buy them. We actually had to make them. And we had to do that because sequencing a genome is hard. So this is what a genome is. One human being, we're composed of about 10 trillion cells. Each cell has 23 pairs of chromosomes. Each pair is about 3 billion bases, so A, T, C, G, the different bases. That data is equivalent to about 1,000 textbooks. And we needed all these machines and robots in this lab of the future because with that technology at the time, we could only read about a few pages a day. So it took us a while to actually make this whole library. But why do we care? And really what we were after is the first genetic map. So before this project, there really was no map. We didn't know how many genes we had, where they were located, what chromosomes, and that's really what we were trying to do. So if you can, this is really the representation. So these are our 23 chromosomes, and it's really a map in the same way that this map of Toulouse shows us where the streets are, where the different interesting locations are. This is a map. In this example are genes that are associated with cancer predisposition, and there are different locations on each of the chromosomes. And if we zoom in, this is one particular cancer gene, and this is printed out. Those are A, T, C's, and G's. And it's associated with, it's in 90% of lung cancers, this particular gene. So we wanted to be able to read that. That's why we created all this technology. And this is an example of knowing where the location, knowing the structure of the gene, we could actually start to look and see what mutations are actually common in patients, what things we could actually find. And so that was so exciting once we had our map and we figured out that we could apply it to all sorts of areas that we decided to create this Broad Institute where I work, where my lab is. And it's the idea that this type of technology, of sequencing technology, genomics, could be applied to lots of different areas. So to cancer, to microbial diseases, human genetics, cancer. And so we formed it as basically this collaboration between MIT and Harvard Medical School and Harvard hospitals with the idea of putting all these different technologies and labs together under one roof to be able to attack all these different areas. And a lot of it was powered by sequencing, by this decrease in sequencing costs that, at the top is that picture I showed you at the beginning of our first genome sequencing center. And 15 years later, this continued advance, it's 100,000 times cheaper than it was at the beginning of 2001 when we finished the first copy. And that's amazing, right? I mean, not even semiconductors have actually progressed at that rate. But it was enabled by this ecosystem. So our lab is, we try to keep it at the leading edge, but it's really powered by this large ecosystem between companies that make the reagents, the sequencing companies, sample preparation, and so as one bottleneck, for example, in reading the DNA was broken, you had to then, the bottleneck might go to sample preparation, so actually taking the cells out of a patient, processing them so that they'd be on a sequencer. And it's this idea that smart labs, labs of the future, really are not, they have to be connected. They have to be able to access ecosystems, to bring in technology from different areas, to partner with even different institutions and do it fluidly and easily without a lot of transaction costs, without having to be burdened by having to reach out to other technologies. This amazing decrease in sequencing costs has enabled us to attack lots of areas. And so the purpose of the Institute is really to understand our own biology at a molecular level, so really to understand our own disease biology at a molecular level across lots of areas. So I showed you genetics of cancer as an example, but it turns out that as our technology got better, we were able to look at other things. You might say, well, how is it possible that even though we have the same DNA in every single one of our cells, how is it possible that I have a skin cell and a hair cell and a muscle cell? How are those things different? And it turns out that there's basically there's the DNA, but on top of it is this epigenetic code that actually allows us to turn genes on and off individually in cells. And so here's an example where we've colored these switches, if you will, these ways to turn genes on and off of twins. So one twin is green, one is red. And so if they're both the same, then it's yellow. But if they're different, they're red and green. And so that's how, for example, twins, maybe one led a very good life doing exercise and having a good diet, and the other one went out and partied and had a good time. Over time, there will be these differences, even though they have exactly the same DNA within their cells. As we were able to decrease the sequencing costs, we could actually start to sequence all of the bacteria. So it turns out that we're composed of about 10 trillion cells, but we carry with us about 100 trillion cells. These are bacteria in our gut, on our skin, and as we were able to sequence and read these cells, they were associated with all sorts of diseases. And so now as we apply this technology, we're starting to find the implication in all of these diseases. And so what we want to get after is basically an engineering approach, and apologies to any Airbus folks, this is Boeing 787, but it's the same idea, I think, in both companies, that you want to have very careful understanding of your system. So to monitor the oil pressure continuously in the engines and monitor the stress on the wings continuously. We want to get to that same level of testing and engineering detail on our own bodies. But there are still lots of challenges. As our sequencing has gotten cheaper, we're now able to actually sequence individual cells. And cells, for example, in cancer, in a tumor, they're actually not exactly all the same. There's differentiation between different ones. And so in order to understand that differentiation, these differences, we needed technologies to be able to actually isolate them and take them out individually. And this is an example of something that we might typically use in a lab, this 96-well plate. The volumes of reagents and cost of doing each one of those assays in each one of those wells is actually quite high. And for a tumor that might have 100,000 cells, that's extremely expensive. So we got together with physicists that are soft matter physicists that had invented these amazing technologies that basically allowed you to make engineered emulsions. And so these, basically what I'm showing you here is there's a chip, so like kind of like an electronics chip. Each one of those little droplets is a droplet the same way that you'd find in an emulsion. The fluid is liquid silicon, and basically we've been able to encapsulate each of the assays that we would normally do in a much larger plate, but now in these little droplets that are picoliter in size, in volume, that are 10 million times smaller. This is slowed down. This is running at about 1,000 hertz normally. And so now with this type of technology, we can much more efficiently access that diversity in a tumor. This technology led to several startups coming out of my lab and co-founded with other labs. The technology came out of Harvard, ESPCI in Paris, and my own lab. And these two in particular are based in Paris, located at the incubator at ESPCI. The first one uses the same technology to identify individual B cells that are the cells that produce antibodies within our body. So when you get a vaccination, these B cells then react to that and produce individual antibodies. So with this technology, we can read that library, if you will, of B cells that contain antibodies and produce therapeutic antibodies that can then become drugs. And the other one does something similar, but for natural enzymes that might be used in food products and other things like that. So just an example of the different disciplines coming together. And let's switch gears a little bit and talk about the other piece, so reading DNA, printing DNA. And so as we've been able to sequence, as we've gotten much better at sequencing, we've been able to read all of nature's recipes. So we can read all these amazing instructions to make all these very interesting molecules. And what we want to be able to do is read these, but actually re-engineer them to use them for our own purposes. And so for that, we're actually making genetic parts. So we're basically making a programming language for cells that incorporate our own programs, if you will, that go into natural systems, but then allow us to reprogram this. And there's just amazing applications. For example, we talked a little bit earlier about how we have all these microbes that coexist with us. So one very interesting project is we're actually sequencing all of the bacteria in our gut and seeing what molecules they produce. So reading all of these recipes that are encoded in these microbes. Other projects are, for example, the pathogens, developing antibodies for pathogens that we can actually do very efficiently. And it's this idea of actually accessing nature's recipes to be able to re-engineer that and use it for our own purposes. So I've shown you kind of a very multidisciplinary view of what a lab of the future is, and if you imagine sort of human knowledge is everything in white, and the leading edge of labs like here in France and other places is that. In my own lab, trying to push a little bit and just trying to make that little dent into the unknown, and trying to push a little bit. Every so often you come across something like the Human Genome Project that then really pushed far into the unknown. But I think a lot of the labs, and I think the common theme is that you have all these other disciplines that are also working very hard to push their own frontier into the unknown. And a lot of what I think has been the experience of the last 15 years in genomics is that you need for all of these things to work together. You need all these disciplines to certainly push in their own direction, but also to work together. And that's really this idea that synthetic biology is this, you needed the molecular biology and the sequencing and all that to push and do its own thing, electrical engineering, programming, soft matter physics. But when you had all of those and you start to connect these things in cool, interesting ways, you get new, really interesting fields like synthetic biology. To do that, labs of the future, I think smart labs, they have to be connected. They have to be aware of the developments in other areas, not just their own, and they need to push hard in their own area, but also in other areas. They need to be multidisciplinary, and you have to be fearless. So it's actually, it's hard and it's stressful to be right on that blue edge, but you have to be fearless. You have to be willing to push into that future. Thank you very much. Questions?